Welcome to the deep dive. Today we are unpacking the foundational blueprint of the human brain. And if you're listening, I know you're the learner, you are insanely curious, and you're looking for that high-density knowledge without all the overwhelm.
Yeah, absolutely.
So we have a stack of incredibly dense, cutting-edge research today. The mission is massive. We are mapping out the human fetal cerebellum, literally cell by cell.
Right.
Our source material is a highly detailed 2024 paper from Cell Discovery by Yang and colleagues. They did a single-cell multi-omics analysis of lineage development and spatial organization in the human fetal cerebellum, specifically between gestational weeks 13 to 18. So, okay, let's unpack this, because that title alone is just a massive mouthful.
It really is.
But the actual science here is essentially the ultimate reverse engineering of human neurobiology.
Yeah, what's fascinating here is that for a very long time, we viewed brain development through a relatively static lens, almost like looking at a finished building and trying to guess how the construction crew put it together.
Right.
But looking at the cerebellum between gestational weeks 13 and 18 is like being handed the architect's live master plan right in the middle of the most chaotic phase of construction.
Wow.
It's a critical window. You have raw, unspecialized progenitor cells rapidly multiplying, making permanent decisions about their identity, and physically migrating across the brain to their final destinations.
Just moving around and set up shop?
Exactly. And by leveraging incredibly advanced tools like single-cell RNA sequencing and spatial transcriptomics, the researchers essentially tagged along for the ride, tracking individual cells as they built this structure from the ground up.
I want to start with a statistic from the paper that absolutely floored me, because it fundamentally changes how you look at the brain. When most people think of the brain, they picture the cerebrum, right? That large, wrinkled outer layer that takes up most of the space in our skulls.
Sure.
But according to this research, the cerebellum, which sits quietly tucked away at the very back and bottom of the skull, occupies over 80% of the total neurons in the human brain. It's staggering.
80%. It's this incredibly dense, highly folded little package. And for the longest time, textbooks basically relegated it to being the body's motor control center.
Yeah, just the balance center.
Right, the thing that stopped you from falling over when you walked. But this paper emphasizes a massive shift in that thinking. The cerebellum is coordinating so much more than just physical balance. It's deeply involved in cognition, emotion, and language.
It represents a profound shift in our understanding of the nervous system. The cerebellum is essentially a massive, high-speed computational engine. It processes complex timing and sequencing.
Okay.
So when it comes to motor control, if there is an impairment in the cerebellum, we see physical clumsiness, a lack of physical coordination.
Makes sense.
But because we now know it also heavily wires into our cognitive and emotional centers, damage or abnormal development here leads to a phenomenon neurologists call the dysmetria of thought.
Dysmetria of thought. That is such a striking phrase. It implies a sort of cognitive clumsiness, right? That is the perfect way to describe it. Just as your body might overreach for a cup of coffee, if your motor coordination is off, your mind might overreach in a social situation or struggle to regulate an emotional response.
Wow.
The source specifically notes that reduced gray matter in the cerebellar cortex is regularly observed in individuals with autism spectrum symptoms or attention deficit disorders.
So it's about timing.
Exactly. The cerebellum regulates the rhythm, timing, and smooth execution of our thoughts and feelings, just as it does our footsteps.
Which makes understanding exactly how it's built so crucial. So let's look at the architectural blueprint. The mature cerebellum has a very strict, highly organized 3-layer map.
Right.
On the outside, you have the molecular layer. In the middle, you have the incredibly unique Purkinje cell layer. And on the inside, the granule cell layer.
3 distinct layers.
But to get to that mature state, the neurons have to be manufactured somewhere. The paper explains that they come from 2 completely distinct germinal centers. And I like to think of them as 2 rival cellular factories operating during fetal development.
I like that analogy. These 2 factories are producing entirely different lineages of cells, which will eventually have to work together perfectly.
Okay, so break down the first factory.
The first factory is called the ventricular zone, or VZ. The VZ is responsible for producing the GBERgic lineages.
And GBERgic means inhibitory, right?
Correct. In neuroscience, these are the cells that act as the brakes in the neural circuit. They tell other neurons to calm down and stop firing.
Got it.
And the absolute crowning achievement of this VZ factory is the Purkinje cell.
And Purkinje cells are visually stunning. If you ever see a microscopic image of one, they don't look like standard neurons at all.
Not at all.
They have these massive, incredibly elaborate dendritic trees that fan out like flat coral reefs or giant sea fans.
They are magnificent structures. And their shape is entirely dictated by their function because they need to catch thousands of incoming signals.
Right.
But to get to their final position, these Purkinje cells have to undergo radial migration. This means they move in a relatively straight line outward from the VZ factory to form that middle Purkinje cell layer.
Just a straight shot out.
Exactly. But to help them do this, the VZ factory also produces something called Bergmann glia. Think of Bergmann glia as the biological scaffolding.
Okay.
There are specialized support cells that extend long fibers, and the migrating Purkinje cells essentially wrap around these fibers and climb them like a ladder to reach their destination.
So the VZ factory is building the brain's heavy-duty brakes and the ladders to put them in place.
Precisely.
Now, on the other side of town, we have the second factory, the upper rhombic lip, or URL. If the VZ builds the brakes, the URL builds the gas pedals.
Right.
It produces excitatory lineages, primarily the granule cells, and these granule cells make up the vast, vast majority of that 80% neuron count we talked about earlier.
Billions of them.
They are tiny, but there are so many. The URL also produces other excitatory cells like unipolar brush cells or UBCs, so you have these two specialized manufacturing plants, one churning out intricate inhibitory control units, and the other just mass-producing these excitatory processing chips.
And the computational power of the human brain relies entirely on the delicate, highly choreographed balance between those two opposing forces.
It's all about balance.
The spatial organization, the precise timing of when these cells are born, and how they physically migrate to interweave with each other is a masterpiece of biology.
So if they don't sync up,
if the ventricular zone and the upper rhombic lip do not synchronize their production lines perfectly during those early gestational weeks, the downstream effects on the brain's circuitry can be severe, leading to those neurodevelopmental issues we mentioned earlier.
Okay, we are about 5 minutes in, so let's do a quick recap.
Good idea.
First, the cerebellum holds 80% of the brain's neurons. Second, it has 3 mature layers. Third, it's built by 2 main factories, the VZ making the Purkinje brakes and the URL making the granule gas pedals.
Perfect summary.
So, considering that delicate balance of emotion and movement we just discussed, I want you, the listener, to think about this. Which of these 2 factories do you think has the harder job? Is it the one building the intricate, massive Purkinje brakes, or the one responsible for mass-producing billions of granule gas pedals?
It's a great question to frame what comes next, because the journey of those gas pedals, the granule cells, is one of the most remarkable processes detailed in the paper.
Yeah, tell us about that journey.
Unlike the Purkinje cells that take a relatively straightforward, direct radial path outward along those glial ladders, the granule cells take a much more complicated multi-step journey once they step out of the upper rhombic lip factory. It all begins with something called tangential migration.
Tangential migration. I was reading this section and picturing an epic, multi-generational cross-country road trip.
That's a good way to look at it.
The granule cell progenitors don't just walk out the front door of the URL and settle down next door. They migrate tangentially, which means they sweep across the entire outer surface of the developing cerebellum.
Right.
They spread out like a wave covering a shoreline. And as they do, they form a temporary structure called the external granular layer, or EGL. It is almost like they set up a secondary pop-up factory right on the surface of the brain.
And that temporary staging ground is vital for building up their sheer numbers.
How so?
In the outer region of this EGL, the progenitors are in a highly proliferative state. They are rapidly dividing and multiplying, essentially cloning themselves to ensure there are enough processing chips for the final adult cerebellum.
So they're just copying themselves constantly.
Yes, through the cell cycle phases, G1S, G2, and M. But once a cell has divided a specific number of times, it reaches a postmitotic state.
Meaning it's done dividing.
Exactly. Its division phase is over, and it is ready to mature. At this point, it shifts slightly inward to form the inner EGL. But the journey still isn't over. Once it is fully postmitotic, it has to undertake a completely different type of movement. It stops migrating across the surface and begins migrating inward.
A deep dive.
Yes, diving deep down past the established layer of Purkinje cells to finally settle in the internal granular layer, the IGL, which becomes its permanent home.
It's an incredible physical journey. Outward, across the surface, multiplying like crazy, and then diving deep back inside. But what makes this 2024 paper so groundbreaking is how they observed this.
They use some incredible tools.
Right. They didn't just look through a traditional microscope and watch cells moving around. They tracked this epic road trip using single-cell multi-omics, reading the genetic diary of each cell along the way.
Cell cycle scoring.
Yes. Through cell cycle scoring, they could look at the exact genetic switches turning on and off at every step of the journey.
And the sheer resolution of modern spatial transcriptomics is astonishing here. The researchers identified very specific genetic signatures that act like progress markers.
What kind of markers?
Well, when the granule cell is in its chaotic proliferative state in the outer EGL, actively going through the phases of cell division, its genetic profile is dominated by the expression of specific markers, notably SFRP1 and ATOH1.
Okay, SFRP1 and ATOH1.
These act as the molecular engines driving multiplication. But the moment the cell stops dividing and transitions to that mature postmitotic state to begin its deep inward dive, those proliferation genes are abruptly shut off.
Just flip like a switch.
Completely shut down. In their place, a completely different set of genes turns on, specifically NHLH1 and STMN2.
Okay, this is dense, so I've got a mnemonic for the listener to help trigger that memory.
Let's hear it.
SFRP starts the fire. That's your proliferative multiplying stage. And NHLH nails it down. That's your postmitotic mature stage.
SFRP starts the fire, NHLH nails it down. I love that. It perfectly captures that biological changing of the guard. The multiplication engine shut down, and the maturity markers kick in to lock the cell into its final form.
And what's amazing is that the researchers didn't stop at just identifying those gene names. They wanted to know what those maturity genes were actually physically doing.
Right.
To figure that out, they ran a gene ontology, or JUCO, enrichment analysis.
A gene ontology analysis is essentially a massive computational method. It's used to figure out the real-world biological processes that these active genes are driving.
So you aren't just looking at the code, you were looking at what the code builds.
Precisely. And the GGON analysis confirmed that in those postmitotic cells, the ones expressing the NHLH1 maturity markers, there is a massive spike in axon development activity.
Axon development.
Yes, this is the crucial proof that neural differentiation isn't just a conceptual label.
Right, they aren't just sitting there looking mature. As they begin that deep dive inward past the Purkinje cells, they are actively growing their axons, the long communication cables of the nervous system.
Spooling them out as they go.
It is like they are spooling out fiber optic cable behind them as they make their final descent, ensuring that once they reach the bottom, they are already wired into the broader network.
It's an elegant system.
So we're at the 10-minute mark. Time for another quick recap.
Let's do it.
Granule cells migrate outwards to the surface, multiply, and then dive deep inwards. And second, specific genetic tags like SFRP1 and NHLH1 track their exact life stage from multiplication to maturity.
Spot on.
So, to you listening, imagine tracking your own life phases with a simple genetic tag. What would your current marker be? Are you in a highly proliferative, chaotic phase of growth? Or are you postmitotic, nailing down your foundations and spooling out your long-term connections?
It's an interesting parallel between cellular development and personal growth.
Definitely.
But as we look deeper into how these genetic tags work across the physical space of the brain, we have to address a major discovery in this paper. One that upends a long-held assumption in evolutionary biology.
Oh, yes. Here's where it gets really interesting. For decades, the working dogma in neuroscience was that the fundamental architecture of the cerebellum is strictly conserved across species.
It was treated as a given.
Right. The assumption was pretty straightforward. A mouse cerebellum develops essentially the exact same way a human cerebellum does, just on a much smaller scale.
Right.
But this multi-omics analysis revealed a massive, undeniable difference between us and mice.
To fully grasp the magnitude of this surprise, we have to understand how genes are regulated across physical space.
Yeah.
The researchers mapped out gene regulatory networks, or GRNs.
GRNs.
Yes, and they mapped them along the anterior-posterior axis. Imagine you are looking at the cerebellum from the very front, the anterior, to the very back, the posterior.
Okay, front to back.
In the human fetal cerebellum, they found that specific sets of genes serve as regional master switches. In the anterior front region, one specific set of transcription factors acts as the core regulators. Genes like RORB, RORA, and ESRG.
So they control the front.
Yes. But as you move to the posterior back region, a completely different network of genes takes over. Factors like TLX3, ETB5, and GLI3.
So they are essentially assigning microscopic zip codes to different regions of the brain. The genes at the front speak one language and the genes at the back speak another.
Exactly. But the real curveball involves a specific gene in this regulatory network called PRM1.
The PRM1 flip.
Yes. When the researchers mapped the spatial distribution of the PRM1 gene in the developing human brain, they found it was heavily enriched in the posterior lobes, the very back of the cerebellum.
Right.
But when they cross-referenced this map with the existing mouse data, the PRM1 gene was enriched in the anterior lobes.
It is completely flipped.
It exhibits a completely reversed spatial distribution across the two species.
Yeah.
And the PRM1 flip wasn't an isolated anomaly. The researchers noted that entire cohorts of genes related to spatial distribution along this front-to-back axis in humans showed almost no correlation with the mouse data.
That's a dramatic, fundamental cross-species difference in how the brain organizes itself.
Huge difference.
And to figure out why this flip exists, the researchers used two incredibly advanced mapping techniques, stereosec and scat-tac-sec.
Right.
Now, stereosec is what gave them the high-resolution spatial zip codes. It tells you exactly where in the brain a gene is turned on. But scat-tac-sec is what really blows my mind because it looks at the physical, three-dimensional structure of the DNA itself.
That is a revolutionary aspect of scat-tac-sec. Traditional RNA sequencing simply tells us which genes are active. But imagine the genome as a massive, tightly coiled library of blueprints.
Okay.
If the library is closed, the blueprint cannot be read. Scat-tac-sec measures chromatin accessibility.
Chromatin accessibility.
Yes. It literally maps out which sections of the tightly coiled DNA are open, physically accessible, and ready to be read by the cell's machinery. By mapping this open chromatin, the researchers could locate specific enhancer regions in the genome.
And what do the enhancers do?
If we connect this to the bigger picture, these enhancers act as local modulators. They dictate the why a gene like PRM1 is physically accessible and turned on in the back of the human brain, but only accessible in the front of a mouse brain.
And these spatial zip codes get even more granular. They didn't just look front to back, they looked side to side.
Yes, the medial lateral axis.
Right. The cerebellum has two large hemispheres on the outer sides and a distinct central column structure called the vermis. The researchers discovered that granule cell progenitors developing in the outer hemispheres uniquely express a specific gene called PRR35.
PRR35 in the hemispheres.
Meanwhile, the exact same type of progenitors sitting in the central vermis are heavily expressing a completely different gene called EBF2.
It is literally like discovering that twin cells born in different neighborhoods are speaking entirely different genetic dialects just because of where they live.
That's incredible.
Which is an absolute breakthrough for understanding localized developmental anomalies. If a neurodevelopmental disorder specifically impacts the central vermis but leaves the outer hemispheres intact, researchers now know they don't have to look at the entire genome.
They can narrow it down.
They can zero in closely on the regulatory networks surrounding EBF2. The level of spatial and genetic resolution provided by this multi-omics atlas is unprecedented.
So recap time. We've mapped out these complex genetic zip codes, GRNs, that assign unique identities to different parts of the brain. And we've uncovered this massive evolutionary curveball where humans and mice have completely reversed spatial layouts for vital genes like PRM1.
Exactly. It fundamentally challenges how heavily we can rely on animal models. If the spatial gene expression in a mouse is completely reversed from a human, a drug tested on a mouse cerebellum might behave entirely differently in a human patient.
Does this massive biological flip make you reconsider how heavily we rely on mouse models for human brain research? It certainly forces us to question our assumptions.
It is one of the most pressing questions in translational neuroscience right now. And it brings us to the ultimate so what of this entire deep dive.
Yeah, why do all this?
Why did these researchers spend countless hours utilizing the most expensive computational tools on Earth to map out a fetal cerebellum at a single cell level? The ultimate value lies in clinical application.
Right.
By creating this multi-omics atlas, researchers have essentially stolen the human brain's proprietary manufacturing blueprints.
They didn't just map the factory, they stole the exact schematics for how to build the machines inside it.
Exactly. We now know the precise transcription factors, the exact genetic markers, and the microenvironmental spatial requirements needed to build specific cerebellar cells.
What does that mean for patients?
This means that in the very near future, scientists could potentially leverage these exact gene regulators in vitro in a highly controlled laboratory setting to grow healthy, customized granule or Purkinje cells from a patient's own stem cells.
That is mind-blowing.
For patients suffering from severe neurodevelopmental disorders or degenerative diseases that cause the cerebellum to atrophy, this atlas is the foundational roadmap for potential cellular replacement therapies. We are moving from observing the brain to possessing the instruction manual to repair it.
It is taking the ultimate mystery of human biology and turning it into an open-source manual for healing. That is the true power of this kind of research.
It really is.
Well, you, the listener, have stuck with us through incredibly dense thickets of neurobiology, genomics, and spatial transcriptomics today.
It was a lot of ground to cover.
It was. We unpacked the rival cellular factories building our cognitive brakes and gas pedals. We rode along on the granule cell's epic fiber optic spooling migration across the surface of the brain. And we uncovered evolutionary secrets hiding in our genetic zip codes.
A very successful deep dive.
But before we sign off, I want to leave you with one final provocative thought to mull over. The authors of this paper explicitly noted that the functional interpretation of that PRM1 gene flipping between humans and mice remains completely unknown.
We have the map, but not the meaning.
Exactly. We have mapped the flip, but we have absolutely no idea why evolution reversed it in humans. But think about this. We established at the top of the deep dive that the cerebellum doesn't just coordinate our physical balance, it regulates the rhythm of our complex human cognition and our deepest emotions.
The dysmetria of thought.
Right. Could this tiny reversed spatial gene pattern, this microscopic flip in our developmental blueprint, be one of the hidden clues to what makes human consciousness and emotional depth fundamentally different from a mouse's?
That is a profound question.
It is a profound, beautiful mystery sitting right there at the back of your skull. Ponder that until our next deep dive.